Welcome back, students. I know that you are eagerly waiting to know about the cardiac cycle. But before that, we'll discuss more about the transporters of our blood. What are they? They are nothing but our blood vessels. In our body, majorly there are two types of blood vessels. They are arteries and the veins. When you observe these tubes, which one is an artery and which one is a vein? This is an artery and this is a vein. Let us observe the structural differences between arteries and the veins. This is the transverse section of an artery and this is the transverse section of a vein. Observe the structure carefully. There is a thick muscular wall in the artery. On the other hand, veins have thin muscular walls. Do you know why? The reason for the thickness of arteries is the oxygenated blood from the heart is pumped with a tremendous pressure. To withstand that high pressure, arteries have thick muscular walls. Now, the pressure in arteries is high when compared with the pressure in veins. Pressure here is low. When you observe the lumen inside the arteries have a small lumen, but veins have a large lumen. Now, there are valves present inside the veins. Then what about arteries? They do not require valves. The heart pumps the blood with a high pressure and there will not be any backward flow of blood in case of arteries. So, valves are absent in arteries and present in veins. Observe the picture carefully. Here, a person is taking an intravenous injection. You might have observed after taking an intravenous injection, there will be a depression seen at the site of the injection. Do you know why? The veins, they cannot retain the shape when blood is absent. They collapse. On the other hand, the arteries, they can retain their shape when the blood is absent.
Veins have larger lumen than arteries. Why?
Reason for the wider lumen in vein is when the blood flows in the veins, it moves in a slow manner. Sometimes it moves against the gravity. To prevent the backward flow of blood, valves are also present. The wider lumen helps in the easy movement of the blood in veins. Arteries resemble a tree in its structure, isn't it? Now, the systemic artery arising from the heart divides into different arteries. Each artery divides into arterioles. Now, these arterioles redivides into tiny microscopic vessels called as capillaries. Suppose the artery from the heart is carrying oxygenated blood and the nutrients to the organ. For suppose take stomach here. Now, the oxygenated blood and the nutrients are used in the stomach. That is, each and every cell in the stomach uses oxygen and the nutrients. Now, the deoxygenated blood moves into the capillaries. From there, it reaches the venules and from the venules, it reaches the main vein. It is how the arteries and the veins function. Now, the bridge between the arteries and the veins is carried by capillaries. Now, the arteries they end in capillaries. The veins, they start from capillaries. The pressure inside the arteries is very high. It moves without valves in the form of spurts or pulsations. The flow of blood is like a pulse. That is why we measure the pulse at the artery only. In the previous class, we have studied the functional differences between the arteries and the veins. Arteries carry oxygenated blood, that is oxygen-rich blood from the heart to all the body parts. Now, what is the artery which carries deoxygenated blood? It is the pulmonary artery. The word pulmonary indicates it is related to lung. Now, on the other hand, veins are the blood vessels which carry deoxygenated blood. That is oxygen poor blood from the body parts to the heart. Based on these differences, let us try to answer the table given in the textbook.
Differences between arteries and veins. Thickness of walls. Arteries have thicker muscular walls. Veins have thin walls. Valves are absent in arteries, present in veins. Capacity to retain shape when blood is absent. Arteries can retain the shape. Veins, they collapse. Direction of blood flow. In arteries, it is from heart to organs. In vein, it is from body organs to heart. Pressure in the vessel. In arteries, the pressure is high. In vein, the pressure is low. Type of blood transported. Arteries transport oxygenated blood. Veins transport deoxygenated blood. Type of blood carried by pulmonary artery. It carries deoxygenated blood. Type of blood carried by pulmonary vein. It carries oxygenated blood.
Let us study another type of blood vessels. They are the blood capillaries. Now, who coined the term capillaries? Yes, Marcello Malfighi coined the term capillaries based on their tiny hair-like structures. These are microscopic blood vessels which are made up of single layer of cells. They can allow or they can diffuse various substances. The leukocytes or the WBC cells, they can squeeze out of these capillary walls. And these blood vessels, they form a continuation between the arteries and the veins. Now, let us perform another activity to know how our arteries and veins are functioning. In the first activity, let us sit in a crisscrossed position or place a knee on the top of the other. Sit for a while. After that, you can observe prickly sensation or the sensation called pins and needles on the top leg. That is due to the restricted flow of blood in the vessels. Now, let us perform another activity. Take your arm and rotate it several times and place it vertically downwards. Now, press your vein in the opposite direction, that is towards the hand. After some time, you can observe a swelling in the vein. That is, we are pressing the vein in the opposite direction of the blood flow. The heart starts to beat at the 21st day during the gestation period, that is during the embryonic development. From the 21st day itself, it starts pumping the blood. That is the reason we all are alive. If it stops working, then there is no more life in us, isn't it? Now let us study the sequential events occurring in the cardiac cycle. When I name it cycle, it means there is a sequential steps, that is continuous steps involved or continuous phases involved in this cardiac cycle. Mainly we study about two different phases called as systole and the diastole. Systole means contraction, whereas diastole means relaxation. So one contraction and one relaxation of the auricles and the ventricles together constitute one cardiac cycle. Let us imagine that all the four chambers of heart are in relaxed phase, that is they are in joint diastole. Now the precaval vein and the postcaval vein brings blood into the right atrium. Similarly, the pulmonary veins, they bring in blood into the left atrium. Now the atria are filled with the blood. The ventricles are still in relaxed phase. Now the auricles enter the auricular systole phase, where the walls of the atria starts to contract. As a result, the pressure inside the auricles is high. There is a pressure gradient in between the auricles and the ventricles. Here the pressure is low. Now the blood will move into the ventricles. Now ventricles accepts the blood. The auriculoventricular apertures is guarded by valves called as AV valves or tricuspid and the bicuspid valves. When blood is in ventricular region, to prevent the backward flow of blood, they closes with a sharp sound called love. That is the first heart sound. The auricles are in relaxed phase. Now the blood will move into the ventricles. Now the ventricles will enter into a phase called as ventricular systole phase or ventricular contraction phase. Here the walls of the ventricle starts to contract. The volume inside the ventricles decreases due to increased pressure. The AV valves are closed. Where will the blood move now? The blood in the right ventricle moves into the pulmonary artery and the blood in the left ventricle moves into the systemic aorta. Now the ventricles will enter into a diastole phase or they start to relax. The pressure inside the ventricles falls down. Due to decreased pressure, there is a chance for the blood in the arteries to come back. To prevent the backward flow of blood, there are valves present at the openings of these pulmonary artery and the systemic aorta. Those valves are called as semilunar valves. Semi means half. Lunar means moon. So there are half moon shaped valves present at the openings of pulmonary artery and the systemic aorta. So these valves closes to prevent the backward flow of blood. So that closure produces the second heart sound, that is the dub sound. So the closure of the AV valves produces the first heart sound, the lub sound, and the closure of the semilunar valves present at the ends of the pulmonary artery and the systemic aorta produces dub sound. Together they form a sound called as lub dub. Time durations of 3 different phases. For joint diastole, it is 0.4 seconds, and for auricular systole it is 0.1 second, and for ventricular systole it is 0.3 seconds. When you total these, it will be 0.8 seconds. So to complete one cardiac cycle, it takes approximately 0.8 seconds. How rapid is our heart? It completes one cardiac cycle in less than a second. Now these phases, they occur one after the other continuously. Joint diastole ends, auricular systole begins. Auricular systole ends, ventricular systole begins. And after the ventricular systole ends, again joint diastole phase begins. So it is a cyclic process. That is why this is called as cardiac cycle.
When you multiply the time taken for one cardiac cycle, that is 0.8 seconds, with heart rate, that is 72 beats per minute, it gives you approximately 60 seconds.
Students, this is the working model of heart in our school. Let us see how it works. Are you able to listen to a sound? What is it? Lub dub, right? Exactly, it is how our heart makes the sound lub dub.
Students, do you know which animal has the biggest heart? The answer is blue whale. Its heart is as big as a car, which beats less than 7 times a minute. Interesting facts about heart. Heart beats 1 lakh times and pumps 7,200 liters of blood in a day. Heart pumps 1 ml of blood from heart to foot, which is approximately 2 meters in just 60 seconds. Through diffusion process, it takes more than 60 years to cover the same distance. Heart continues to beat even outside the body due to its own electrical impulse for a period of time with adequate supply of oxygen. We usually say that a heart skips a beat when we sneeze, but actually, sneezing doesn't stop your heart from beating. The most important thing is laughing is good for your heart.
Now you have understood how amazing is our heart, right? We need to keep our heart healthy by following healthy lifestyle and healthy food habits. Fishes are considered as superfood for your heart. It contains omega-3 fatty acids. Let us stay there at the fish and observe how its heart chambers look. It has only 2 chambers, one atrium and one ventricle. It is an example for single circulation. Let us observe the differences between single circulation and the double circulation. Blood flows only once through the heart to complete one cardiac cycle in case of single circulation, but whereas in double circulation, blood flows twice through the heart to complete one cardiac cycle. Now in single circulation, heart is only two chambered. It has only one atrium and one ventricle. In double circulation, the heart is four chambered. That is, the upper two chambers are called as auricles, the lower two are called as ventricles. Venous blood flows through the heart in case of fishes. Venous blood is nothing but deoxygenated blood, and in double circulation, you know, two types of blood flows through the heart, that is deoxygenated as well as oxygenated blood. And single circulatory system is less efficient when compared to the double circulatory system.
Let us see how the transport system has evolved in different phylums. In protozoans like amoeba, the protoplasm shows Brownian movements, because of which the nutrients and oxygen are distributed. Phylum Porifera. Water canal system is seen in sponges. Phylum Cnidaria. Jellyfishes have developed blind sac-like gastrovascular cavity which transport nutrients to each and every cell. Phylum Platyhelminthes. Example, Fasciola hepatica, liver fluke. Branch digestive system supplies nutrients to the cells directly. Phylum Nemathelminthes. Example, filarial worm. Pseudocoelom collects and distributes the food materials. Phylum Annelida. Example, earthworm. Closed circulatory system is seen in annelids. That is, the blood flows in blood vessels. Pulsatile vessels, dorsal and ventral vessels are seen in earthworm. Phylum Arthropoda. Example, cockroach. Open circulatory system is seen in arthropods, which means blood flows directly to the tissues. Arthropods have developed pulsatile organ to pump the blood to the heart. Phylum Mollusca. Example, snail. In molluscans, open circulatory system is seen. Some cephalopods like octopus have closed type of circulation. Phylum Echinodermata. Example, starfish. Closed circulatory system is seen in echinoderms. Phylum Chordata. In higher animals, closed circulatory system is seen. Heart is divided into chambers. Differences between open and closed circulatory systems. In open circulatory system, blood flows through large open spaces among the tissues. In closed circulatory system, blood flows through chambers of heart and blood vessels. Tissues are in direct contact with blood in open circulatory system. Blood doesn't come in contact with tissue in closed circulatory system. When you compare the efficiencies, open circulatory system is less efficient than closed circulatory system. Open circulatory system is seen in mollusks and arthropods. Closed circulatory system is seen in annelids, echinoderms and higher chordates.
When you travel overnight or when you sit for longer hours, then you can observe swelling in your legs, isn't it? That condition is called as edema. Why do our legs swell?
In edema, tissue fluid builds up in our legs, causing a swelling. What is called as tissue fluid? You know that blood flows through the capillaries to supply nutrients to the tissues. Liquid portion of blood with nutrients flow out of the capillaries. The tissue fluid present in the tissues should be transported into the blood vessels again. Only some portion of tissue fluid enters the vein. Then what about the remaining fluid?
The remaining tissue fluid moves into the main bloodstream by a separate system called as lymphatic system. This lymphatic system is parallel to the venous system. Now the word lymph means water in Latin. Lymph forms an important link between blood and the tissues. Our blood contains both solid and liquid particles. When the solid particles are removed from the blood, then it becomes lymph. So, lymph contains only liquid particles or the fluid particles. How do the lymph flow? The muscles in our skeletal system contracts, due to which the lymph flows in our body. Are there any valves present in the lymphatic vessels? Yes, I have told you the lymphatic system is parallel to the venous system. Both lymphatic system as well as venous system contains valves. What do we usually say when we are angry? We say that our BP has increased, right? BP is nothing but our blood pressure. There is a device called as sphygmomanometer with which you can measure the blood pressure. BP, that is the blood pressure, is the pressure created in the heart due to blood flow in the ventricles of the heart. When the ventricles contract and when they relax, they create a pressure called as blood pressure. In normal adults, the BP is 120 by 80 millimeters of mercury. What does this 120 over 80 indicates? 120 indicates the systolic pressure. And 80 indicates diastolic pressure. If the blood pressure is less than 90 over 60, then that condition is called as hypotension or low BP. If the blood pressure is greater than 140 over 90 mm of Hg or mercury, then that condition is called as hypertension or high BP.
Systolic pressure is the pressure with which blood is forced out of the ventricles during ventricular systolic phase, that is during ventricular contraction. It will be 120 millimeters of mercury. Ventricular diastolic pressure is the pressure observed in ventricles during ventricular diastolic phase, that is when the blood starts to refill the ventricles. It is a lesser value. It will be 80 millimeters of mercury. Usually, doctors measure the blood pressure at the upper arm artery to get standard value using a sphygmomanometer.
What happens when you are injured? Your blood flows, right? What happens after some time? It stops bleeding. What happened there? The blood clotting process has happened. The clotting time of blood is in between 3 to 6 minutes. Now this process of blood clotting is otherwise called as coagulation of blood. After the blood clot occurs, when you touch the injured part, a liquid sticks to your fingers. What is that liquid called as? It is called as serum. Serum is a yellowish fluid present over the blood clot. There are 3 different types of cells in our body. They are RBC, erythrocytes, WBC, leukocytes, and platelets, the thrombocytes. These blood platelets help in coagulation of blood. There are 12 different coagulation factors which help in the process of coagulation. There is a chemistry involved in the coagulation process. As I told you, the blood platelets helps in the coagulation process. they release an enzyme called as thrombokinase. This enzyme acts on the prothrombin present in the blood. This prothrombin is converted into thrombin by thrombokinase enzyme. There is another substance called as fibrinogen, which is in soluble form. Now this thrombin acts on the fibrinogen and converts this fibrinogen into insoluble fibrin form. These fibrin particles, they trap the blood cells and they pull the edges of the wound during the coagulation process. The clotting time is in between 3 to 6 minutes, but in persons with vitamin K deficiency, it takes more time for the blood to clot.
How does vitamin K helps in clotting of blood? Vitamin K makes various proteins like prothrombin needed for blood clotting. During blood coagulation process, platelets release chemical signals and enzymes to attract more platelets. As a result, platelet plug is formed to prevent external bleeding. In addition, clotting factors causes fibrin fibers to stick together and seal the wound. Genetic or inherited blood disorders. Hemophilia, a genetic blood disorder in which blood doesn't clot normally, due to which excessive bleeding occurs after any injury. Thalassemia, a genetic blood disorder in which the body makes abnormal hemoglobin.
Students, I hope you all have understood the transport system in animals. Thank you.